Today we are going to talk about the immunoglobulin isotypes. So you have to know that there are various isotypes of the immunoglobulin. Those are immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, and immunoglobulin E. We call it IgG, IgA, IgM, IgD, and IgE. So in the previous video we have already discussed about the antibody structure and function. Now we are going to talk about the various immunoglobulins isotypes in this video. So before actually explaining the each and every types of immunoglobulins, I want to talk about brief introduction about the immunoglobulin isotypes. So you have to know that all isotypes can exist as monomers, but some of the isotypes may exist as polymers also, like IgA can exist as dimers, whereas the IgM can exist as pentamer. And the mature and naive B cells, prior to activation, they express the IgM and IgD on their surfaces. When the B cells differentiate in the germinal centers of the lymph node by the isotype switching into the plasma cell, they secrete other type of immunoglobulin, that is IgA, IgE, or IgG. So this you have to know that. And I want to discuss brief about the affinity and avidity also. The affinity refers to the individual antibody antigen interaction, while the avidity describes the cumulative binding strength of all the antibody antigen interaction in the multivalent molecule. So this affinity and avidity term in the immunoglobulin you should know. Again I am going to repeat. The affinity of the antibody refers to the individual antigen antibody interaction, but the avidity describes the cumulative binding strength of all antibody antigen interaction in the multivalent molecule. Now let us discuss about individual immunoglobulin isotypes. So first of all, let us discuss about immunoglobulin G. The immunoglobulin G is the main antibody in secondary response to an antigen, and it is the most abundant type of the immunoglobulin. The most abundant type of immunoglobulin in serum is the immunoglobulin G, and it is the only immunoglobulin that crosses the placenta. So it is the only isotype of the immunoglobulin that crosses the placenta. The various functions of the immunoglobulin G are: it fixes the complement, it opsonizes the bacteria, it helps in to neutralize the bacterial toxin and various viruses. So I have already told that it is the only isotype that crosses the placenta, and it provides infant with passive immunity that start to wane after the birth. So this is the brief introduction about the immunoglobulin G. Now let us talk about the immunoglobulin A. So immunoglobulin A, it basically prevents the attachment of the bacteria and viruses to mucous membrane. So you have to know that unlike the immunoglobulin G, which fixes the complement, the immunoglobulin A doesn't fix the complement. So immunoglobulin A doesn't fix the complement. It occurs as the monomer in the circulation, but it occurs as the dimer. You can see the dimer with J chain when it is secreted, and it crosses the epithelial cell by the transcytosis. There is a mechanism called the transcytosis. It crosses the epithelial cells by the mechanism transcytosis. So it is produced in the GI tract. So basically in the GI tract, Pierce patches, there is the structure known as the GI in the GI tract, that is Pierce patches. The Pierce patches specifically produce immunoglobulin A, and the immunoglobulin A protects us against the gut infection. For example, the Xydria. The Xydria is an organism that causes the Xydrosis. So immunoglobulin A basically protects us from the Hydra. And it is the most produced immunoglobulin in our body, but the concentration in the serum of this um, immunoglobulin is very less because it is produced in the gut. And what happens is that it is released into the various secretion. So the various secretion like tears, saliva, mucus, and also in the breast milk, it is secreted. So it picks up the secretory components from the epithelial cells, which protects the Fc portion of the luminal proteases. So you have to know that the immunoglobulin A also can exist as the monomers, but when it is secreted, the J chain is present and it exists as dimers when it is secreted. Now let us talk about immunoglobulin M. The immunoglobulin M, sometimes called as the millionaire molecule, because there are 5 immunoglobulin present in the immunoglobulin M. So first of all, you have to know that the immunoglobulin M is produced in the immediate response to an antigen. So if some antigen we encounter which is foreign to us, and the immediate response in our body is done by the immunoglobulin M. So immunoglobulin M is the first most immunoglobulin production. That means primary response to an antigen is done by the immunoglobulin M. So like immunoglobulin G, it also fixes the complement. And the antigen receptor on the surface of the B cell. So the antigen receptor of this immunoglobulin M is present on the surface of the B cells. It is monomer on the B cell. On the surface of the B cell, it exists as the monomer, but when it is secreted, when the immunoglobulin M is secreted, it exists as pentamer. So you can also see the J chain when it is secreted in our body. So this pentamer enables the avid binding to the antigen while humoral response evolves. So the structure of the pentameric structure of the immunoglobulin M is quite functional. Again, I am going to repeat. The pentamer structure of the immunoglobulin M is quite peculiar because the pentamer enables the avid binding to antigen while humoral response evolves in our body with the time. So now let us discuss about the immunoglobulin D. So the unlike other immunoglobulin, the immunoglobulin D has unclear function, but immunoglobulin D is found on the surface of many B cells in the serum. Now let us talk about the immunoglobulin E. The immunoglobulin E is one of the important immunoglobulins because it causes type 1 hypersensitivity reactions. The detail about the hypersensitivity reaction we'll talk in another video, but the immunoglobulin E has very important functions. So immunoglobulin E binds with the mast cell and the basophils. It cross-links when it is exposed to an allergen, and it also participates in the type 1 hypersensitivity. So it participates in type 1 hypersensitivity means it mediates the immediate hypersensitivity through the release of the inflammatory mediators such as histamines. So it also contributes to the immunity to the parasites by activating the eosinophils. So this is one of the important functions of the immunoglobulin E. Now let us talk about the antigen type and memory. You have to know that there are two types of antigen, and one is thymus independent antigen and another is thymus dependent antigens. In the thymus independent antigens, you have to know that the thymus independent antigen are the antigens that are lacking the peptide component. So thymus independent antigen are those antigens that lack the peptide component. For example, liposaccharides from the gram-negative bacteria. So it cannot be presented by the MHC2 T cells. They are weakly immunogenic, So the vaccine often required boosters and the adjuvants. For example, capsular polysaccharide subunit of Streptococcus pneumoniae, PPSV23 vaccine. So these are the thymus independent antigen. On the other hand, which is the thymus dependent antigen? Those thymus dependent antigens are the antigens containing a protein component. For example, Streptococcus pneumoniae PCV13 vaccines. The polysaccharides conjugated to diphtheria toxin like protein. So these are the two antigens containing the protein components. The another is class switching and immunologic memory occurs as the result of the direct contact of the B cell with T helper cells. So thymus dependent antigens are very much important, and class switching and immunologic memory confers the long lasting immunity in our body. So these are the concept of the antigen type and the memory of our body. So I hope you understood this lecture. Here in this video we talk about various immunoglobulin isotypes. Again, I am going to repeat, there are mainly 5 isotypes of the immunoglobulin. They are immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, and immunoglobulin E. And we also talked about the antigen type and the memory. You have to know that there are two types of antigen: thymus independent antigen and thymus dependent antigen. The thymus independent antigens are the antigen lacking a peptide component, whereas the thymus dependent antigen are the antigens that contains a protein component. So this much for today. Thank you.